You may be wondering what this pet store has to do with the biopharmaceutical industry. Let's go ask this hamster.
Are you in need of proteins but are tired of trying to find fast-growing genetically characterized cells? Are you looking for a cell line that grows quickly in suspension and can be genetically engineered with ease? Well, look no further than Chinese hamster ovary cells, also called CHO for short.
CHO cells are a cornerstone in the biopharmaceutical industry and will certainly meet your protein production needs.
The CHO cell line is a well-established platform for the production of biopharmaceuticals for three main reasons. Over the years, CHO cells have been extensively studied and optimized, making them a reliable system for large-scale protein production. Mammalian cells are necessary instead of bacteria for production of certain proteins due to specific post-translational modifications needed, such as glycosylation for proper protein structure and function. Having protein structures similar to what is naturally found in humans decreases the chances for adverse immune responses. CHO cells can also be cultured in large volumes, which is crucial for commercial scale production. With bioengineering, CHO cells' ability to express complex therapeutic proteins in high amounts is being maximized.
You may be wondering how CHO cells came to be.
Well, CHO cells were first isolated and cultured in the 1950s for use in cell biology research. The CHO cells grew rapidly in laboratory cultures. What a surprise! The cells were resilient, had fast generation times, and chromosomal heterogeneity, which meant chromosomal abnormalities would be apparent. Perfect for research. Once DNA recombination technologies took off in the 1970s, CHO cells were found to be easily genetically modified. These were the first signs that CHO was the way to go in scaled-up operations. In 1987, tissue plasminogen activator used to treat blood clots produced by CHO cells was approved for clinical use, becoming the first recombinant therapeutic protein produced in mammalian cells. In modern times, nearly 70% of all recombinant protein therapeutics produced are made in CHO cells.
How does one care for their CHO? Well, let me show you how to CHO. Taking a look into the CHO's natural habitat, we see our little buggers attached to an anchoring media within our CHO incubator, otherwise known as a bioreactor. Here, the CHO cells grow, thrive, and produce our juicy product.
Look at those CHO go.
To maintain our CHOs, we need to closely monitor our reactor parameters. For optimum conditions, our CHOs must be kept at a 7.2 pH with an impeller agitation below 600 RPM without shear preventative additives. Now, let's not get too hasty with agitation. We don't want our CHOs to have shear stress or worse, attachment issues. Agitation speeds are best determined for the scale of production and application. For CHO growth, we want to maintain a temperature of 37 degrees C, while for protein production, we want a temperature of 32 degrees C. To prevent CHO starvation, we must apply the proper nutrients with adequate concentrations consisting of carbohydrates, lipids, amino acids, and nucleic acids. Use a defined media for accurate cell production. Don't forget to discharge oxygen at approximately 30% dissolved oxygen with 5% enriched CO2 air buffer for adequate health and metabolism. Need to preserve CHOs for later? A temperature of negative 47 degrees Celsius for long-term storage, and for better cell viability at negative 32 degrees Celsius. Need to genetically engineer our CHO? Use the CRISPR-Cas9 system for targeting genome editing in CHO cells, allowing for rapid and efficient engineering of cell lines, and to create new proteins for future needs. Speaking of the future, let's take a look at the possibilities CHO offer.
Want to help treat illnesses?
Pharmaceutical companies need engineers to design and improve upon growth, productivity, and stress resistance of CHO cells. Bioprocessing developments have enabled the expression of high-quality recombinant proteins for monoclonal antibodies, and will continue with your help. Firms need you to design, troubleshoot, and optimize bioprocesses and equipment for CHO cell production. Do you have what it takes to be a CHO researcher? Look at all these opportunities. As you can see, CHOs need you. The more you CHO.
Warning: CHO cells are susceptible to stress. At times, CHO cells may experience limited growth rates and low productivity. Keep your CHO cells happy or they may initiate apoptosis, leading to rapid cell death and decreased protein yields. Remember that your CHO cells exhibit high consumption of glucose and glutamine. Nutrient depletion will occur quite quickly. You may notice speedy accumulation of unwanted products such as ammonium and lactate. These products can further impede cell growth and protein quantity.
Try CHO cells today! CHO, CHO, CHO,